as you see the title, right? The company is called Atlantis AI360, and I'll tell you why that is so. The essence of Atlantis is that it gives you a search engine. And of course, there's like more detail to that. So what it gives the user is a search engine where you can, let's say, put two keywords, and for example, HIV and aging, HIV atherosclerosis. And it allows the user to dive deep into the subject matter, understand the molecular mechanisms, understand the key players, the signaling pathways. And once you get that data, it actually derives insights from that data. So it is more powerful than Google. It is powerful than ChatGPT. I've done some analysis with against ChatGPT as well. With ChatGPT is the rage of the day, right? Everyone wants to do ChatGPT and Google, right? There's no comparison. So that is the genesis to be able to help the workflows of a typical researcher to be able to reduce the grunt work and be able to advance their key research because that's their focus. They don't want to be dragged down into okay, which paper should I read? What does it mean? How do I advance my research? Right. So that's the key pain point that I want to address. Right. And also by doing that, what I've heard in the recent past is that there's so much of focus on researchers getting their own grants, right? Without grants, you can't survive. That's what academia. And beyond just grants, you have to be able to generate funds from any means necessary. You have a novel antibody, you have a novel small molecule, you have a novel protocol, reagent, whatever it is. You should be able to monetize that. But in a university, I'm not sure whether you're able to monetize that. So that's why I mentioned storefront. So Atlantis actually gives you a storefront. So today you say, Sanjay, say, hey, I have an antibody against, I don't know, CXCR4, with a certain phosphorylated molecule, right? It is not on Promega, it is not on Bicopia, it is not on any, portal. But Atlantis will allow you to evangelize that particular antibody to a large research community, and you'll be able to actually monetize your research. It is not just going to be in some journal. It will allow you to monetize that. So that's the beauty of it. So it does both. It provides you a search engine. It also helps you monetize your assets, right? Your IP. So that's the overall focus. And it's all driven by natural language processing. So it takes textual data from abstract, from journal articles, from USPTO, from clinicaltrials.gov, any data set that you want, and derives the insight from that. So that is the overall, just the background of it, right? So let me dive into this. Let me go to presentation mode, right? And let me minimize this, and we can start talking. Yeah, here you go. So right, that's about me. I will skip this, but essentially what I'm trying to say here is that I've got experience from the research side, from the business side, from the management side. I've written 3 books on bioinformatics. So Tata McGraw-Hill, Springer. So got a lot of experience actually building platforms which will allow people to really crunch that data, crunch their raw information into something which is more meaningful, right? Without having to do it manually, because so much of data you can't do it manually. So that's why you need bioinformatics tools to be able to really crunch that data very quickly, and you'll be able to derive insights from that. Okay, so here is the problem statement. If you look at the top left here, so journey of a data user consists of exhaustive literature service because we do that day in and day out. You go to PubMed, you do for do a lot of searches, and then you generate hypotheses. Then you design experiments. Then you validate your hypotheses. You review your findings and you publish your results. All of which is very manual, right? And it is repetitively time consuming. It is very resource extensive, resource intensive. So in life sciences, it's all the more daunting because of the complex, multi-dimensional, multifactorial nature of the data and very high velocity and volume of data. And Google provides access to data, but we don't have a problem with access to data. It is not the problem. The problem is driving insights. So the solution is Atlantis AI. So it brings together automation, NLP-driven generation of insights, complete translational views of the subject matter, and also derives knowledge assets. By which I mean reagents, fine chemicals, enzymes, antibodies, clones, disease models, right? All of that. Even KOLs, patient recruitment side. So it does that as a matter of, if once you do a search, it will look at those assets and serve those assets up to you. And of course, you have the layer of deep domain expertise, because without deep domain expertise, without understanding the domain, there's just no way you can derive, useful information out of the data, right? So Atlantis consists of 4 components. So there's a content automation engine, which is called Genie, which is a search engine. It has got the insight generation engine, which is NLP based, natural language processing, NLPAC. It's got an interactive browser, and it creates a set of storefronts. So users can set up storefronts to then monetize their IP. So that's the overall construct. This is what Atlantis components look like. Now, of course, please feel free to ask questions, but I will, breeze through this fairly quickly. So here is the customer journey of a typical researcher. When I say customer journey, it is typically the interactions that a researcher may have with a PubMed, with any online web resource or a desktop resource. What is the number of steps the researcher takes? What is the output of that? How does he or she utilize the output? And how does that whole workflow start from start to finish, right? So let's say you are a HIV researcher, you go to a lab meeting or to a conference, you read a paper, and you get some, new ideas, and you want to try out, different directions to advance your field. And how do you do that? You want to review your own data, your own findings, saying, okay, how does it connect to what is published? Then you discuss theories with your team, with your co-workers to understand how I can advance my field, right? You want to read about external research, latest journal articles, available disease models, reagents, what's out there, right? How do you leverage that and integrate your findings versus the external findings, right? So could be a new signaling molecule, could be a novel member of a pathway, could be a drug intervention, could be other areas of interest. These are just examples, right? So you go to maybe go to a PubMed or go to individual journal sites where you have access to. You can go to clinicaltrials.gov, maybe other resources that you have. But in the end, what does your workflow look like? Do you download articles? Do you browse online? And how often do you conduct these searches? I mean, do you conduct searches daily, weekly? How do you summarize the findings? Because then you have to read those papers. The papers could be 6 page long, 10 page long. It's a lot of data for you to handle. So how do you summarize these key findings? And how do you retain that data? I mean, once you read a paper, you put it on your desk, you forget about it. What do you do? Is it a soft copy or a hard copy? Even if you maintain hard copy, you'll have, whole stack of papers sitting on your desktop for, forever, right? I mean, how do you retain that data? How do you retain the information? And what does a typical day-to-day workflow look like? So this is, if I assume that this is the typical customer journey of a researcher, then this is where Atlantis AI, comes into picture. So let me pause here and maybe just get a quick overview from you whether this is relevant. This is this is you relate to these pain points, or am I off? Any comments from the audience so far? Hello. Oh, Sanjay, you're on mute. Sanjay, you're on mute. I can't hear you.
Sorry, sorry.
Yeah, yeah, yeah.
No, this makes sense, and this catches the right sense of what we do every day. But
Okay.
one thing I want to point out it's not individuals. It's a team-based science. Multiple
Right.
people are involved, and you
Sure.
have collaborators from different areas of expertise, and
Right.
you should account for that too.
Sure. Sure. Yeah. I mean, of course, it's in the end, uh, it's a team sport. Let's say. Yeah.
Yeah,
I understood. Understood. Yeah. Yeah. Okay. So, any other comments, or should I move on?
yeah. Please go ahead.
Okay, all right. Okay, so the next slide. So this captures the key pain points. So on the top panel you see here, this is actually my advisor at Dana-Farber. He was an MD, PhD tenured faculty. So he said to me, Harsh, I need to manage the research effort of my graduate students, my postdoc in the lab. I'm also a clinician, so I need to manage my patients at the cancer center. I also need to take my exams annually, so I have to do a lot of reading. How do I keep on top of literature? How do I manage my exams? How do I take care of my postdocs? It's a whole big, effort on my side. I don't have any free time remaining. I have to do so much of reading, right? Again, the second example is the PhD tenured faculty at Rochester. This is Steve Dewhurst. He said the same thing. I had to manage students, postdocs. I have to keep on top of literature. I mean, I used to see him read. I mean, I whenever I goes to his office, he had a whole stack of papers, a mile high, and all the latest peer-reviewed journal articles. And then you have to generate data, your insights. How do you support new research directions? You write grant applications. So typically, that is the life of a typical researcher. Could be a faculty, could be a postdoc, could be a young researcher, right? And that's the specific pain point I'm trying to address here. Right? On the bottom of the slide are actual users. So I've tested this out with a number of people in biopharma companies as well as research laboratories in hospitals. These are all PhDs or MD PhDs, and also from Wall Street. And they said Atlantis offers access to scientific data, but also in a persistent format. right? And other resources such as Crunchbase or PitchBook, they may not provide that level of detailed scientific analysis because those are more business oriented. But if you really want to make a decision on whether this drug is really effective or not, or this data makes sense or not, you need to know the science behind it. You just can't focus on the business aspects of how much is this drug worth. But unless you understand the pathway, the mechanisms, you are not able to really make a sense of the value of that. So that was clearly feedback from a researcher from Pfizer. If you look at the right hand side, again, automated retrieval of data from journal articles. I mean, you can go to PubMed, do a search, but then what that, right? You are left with just links, and there is no way for you to retain that information, right? So Atlantis provides you a way to retain the information in a persistent format. So the feedback was it is very useful for both clinical as well as research laboratories. On the bottom again, this is from AZ. So they wanted to understand, is there a gap in their pipeline or in their portfolio where they say, for example, in GI, they're basically on gastric cancers. Do we have enough of a portfolio, either early phase or late phase, or even marketed molecules where they say, okay, the patients are covered for the entire GI portfolio, right? So that was something which I did with JCO from the ASCO journals, and able to understand what gaps are there in the portfolio. Again, Atlantis provides information. So it is, that's the beauty of Atlantis. It is able to look at any subject matter, do a deep dive, extract the insights, and then, build decision trees based upon deep analysis. On the bottom right, it's MD physician, which is who's covering biotech stocks from Bloomberg. And he said, we don't have this. What you showed me is, the automation, the insight generation, also geolocation mapping. So I'm able to tell you exactly where in the whole world is a specific HIV lab or a specific cancer lab that you're interested in, and then you could take that information and build collaborations, to joint grant writing, all the stuff, right? And while they are a data and tech company, it's a big firm, right? It's a well-known firm, but they don't have the domain expertise to really do that cutting edge science that really is needed for doing deep analysis on biotech stocks. So overall, I find that yes, I'm on the right track. It provides the information and value that researchers need. So I'm really excited here to showcase this to a much larger audience with the essential premise that there's a lot of data, and Atlantis really helps you process that data in a more intelligent fashion, right? So again, I will pause, but feel free to stop me for any question that you may have. So otherwise, I typically tend to go very fast. But again, feel free to ask me questions. Okay, moving on. So these are the questions I got from the audience. So can you take a moment to speak directly to the community? How will this work affect? How will this work effort affect the day-to-day life, right? So how is Atlantis useful on a day-to-day basis for a typical research? So that was one question. Second question is, does it allow people to identify new techniques, new research directions? So these are the top two, feedback questions. At the bottom is more related to how Atlantis can leverage the AI and ML space for advancing biomedical research. So I'll handle these two things differently. So the top two questions are pretty fairly straightforward, right? So Atlantis streamlines workflows. So automatic extraction of data from any repository, could be clinicaltrials.gov, could be NCBI resources, PubMed, PMC, all kinds of databases from the NCBI databases, could be individual university websites. All of these are fully automated, can be automated in a customizable fashion. It reduces grunt work. It accelerates the research effort because it gives you those insights. So you focus on deriving hypotheses and actually designing experiments, right? So that should be your focus, and Atlantis helps you get to that level of acceleration. It identifies research assets. Could be a fine chemical, could be an antibody, could be a disease model, could be a zebrafish model that is, hey, this is available at NYU or UCLA. This helps you identify those assets. And also, if you want, like Sanjay, you said, right, this is not just individual work. These are joint R&D opportunities. So you can design a community of scientists who can then work together, write joint applications, write joint grants, and it really helps people to come together to solve a common problem, right? And also now in terms of the organization, so Atlantis helps you organize the information in a PowerPoint slide. In the end, the PowerPoint slide is the most effective way of presenting your data to in front of an audience. You can maintain longitudinal research record of your own internal research, overlay with external findings, and also it gives you web-based dashboards. You're able to do easy, quick point and click access to all kinds of data. So I'll show that to you in a practical demo. But it allows you to persist that data in a easily used format. So PubMed doesn't do that. PubMed, you go and do a search in PubMed, you just get a bunch of links. Then you download the papers, take a printout. What then, right? But Atlantis helps you. It organizes data in a PowerPoint slide. So that's the beauty of it. And in the end, hey, you suppose you're going to a symposium or a conference, or you're doing a journal club. You want to quickly find out what's the latest data about a certain field. You're able to do that in a jiffy. It's very fast. It's blazing fast, and gives you that PowerPoint deck in a matter of minutes. 30, 40, 50 slides in a matter of minutes. So that's how fast it is. So this is overall in terms of how it works, how it affects how it helps your day-to-day basis workflows, and enhances your overall research productivity. Any questions on this? Okay, so I'll move on. The second set of questions is about how to apply AI/ML. Okay, so the questions I would have for the audience is that, okay, what is your end goal? What do you wish to model? Right? What data do you have to support your analysis? What algos can you apply? What algorithm can you apply to train and test your model? How do you refine that model? I mean, how do you ensure accuracy? But how can you validate those results, and then how do you apply them in real life? So, in terms of applications of AI/ML, this is how, I mean, I would typically see, kind of create the whole problem statement. So there is supervised learning, unsupervised learning. I'm not sure how much deeper I need to go into this, but this is kind of a very quick and dirty overview of machine learning. So, there is supervised learning where the data has labels. So, for example, a certain molecule has a certain toxicity and a certain activity against a receptor, and you can utilize that knowledge to then identify the properties of a novel new chemical entity that comes your way. The second is unsupervised learning, where you may not know the relationship between the molecule and the activity, and you can cluster based upon certain similarity in properties. So, for example, structural similarity, chemical similarity. You can cluster those molecules with the assumption that molecules which have similar chemical properties will have similar biological activities also. I mean, this is an assumption. It may not always to hold true, but that is the idea. And then based upon that clustering, you're then able to identify novel candidates for drug development. So that is the overall, machine learning essence of, how do you apply these algorithms for these two kinds of scenarios. And here is exactly how this is done. So you have the objective is I want to model the interaction between a drug and a biological target. Then you collect the data. Data could be molecular structures, physical properties, chemical properties, bioactivity, IC50. Then you determine what are the key features. So you identify binding affinity, which is a very important feature. What is the toxicity? What is the specificity? And utilize that information to do a training set. The training set essentially is given these known molecules, can I create a model where once I've got that model, it will predict in an accurate manner whenever a new molecule with unknown properties comes across. So that is the training or the model training. And then you evaluate the model efficacy and accuracy. Then you validate that data, maybe in a wet lab, and then you apply once your model is perfected, then you are able to then utilize that model to then test on unknown compounds. And that's how the whole application of AI/ML and biomedical research actually works. And Atlantis AI360 can help you do that. Okay, now some quick case study, but I'll go to a different section now. But like a quick pulse check if there are questions from the audience.
So, Harsh, one question I have is that you so far talked about the literature search and
Hmm.
kind of getting the whole ocean worth of information into kind of some PowerPoint slides and all that. But say I have been working with the developing a drug with some medicinal chemistry we are doing, and we end up in having multiple small molecules in our search.
Hmm.
This is something practical. It's not in the PubMed or anywhere on the online. It is in my lab. How I can
Hmm.
integrate that kind of information into this the
Yeah,
application?
sure. So it goes back to this particular slide here. So what you have today is, I think, maybe supervised learning is a good example, the top example, right? So there are known small molecules. I mean, you have a database of known molecules with known structural properties, known chemical physical properties, and a known biological activity. So that is your training set. Now you are saying that I have something which is completely unknown in the public domain, completely new structure. It has a new side chain. Maybe it has a fluorine, maybe it has a chlorine, maybe it has a heterocycle, right? It is had a certain molecular weight, it has certain solubility, lipophilicity, electronegativity, size and shape, all of that information, you may or may not have. But that is a novel molecule. But it is known in nature, and of course, based upon our own research over the last 100, 200 years of pharmaceutical industry, we know that a certain scaffold, a certain nucleus has a certain biological activity, a certain toxicity. So you are able to then model this data, which is available today, right? And now you bring your own molecule and say, okay, this is a novel molecule, molecule X, right? Now tell me, is this molecule X even useful? Is it toxic? Can it be used in a clinical situation? How should I develop this molecule further so I'm able to get the right pharmacological activity, right clinical activity without the toxicity? So I think your answer is fairly, I would say it's a typical problem in drug discovery, because you have known molecules and you have an unknown molecule, and you're able to model the known data and then ask the question. This is the same model that at the bottom you see here, unknown compounds. So you can model the known compounds and then ask the question, what does my unknown compound look like?
Okay.
And you are able to then predict with a fairly high degree of accuracy that your molecule could be active in a certain configuration. So maybe you say, okay, I need to change this fluorine. It is too toxic. I need to change that to a hydrogen, or I need to change this methyl group into something different, right? Maybe add a fluorine there, and that will make it more increase the affinity to the target. So all of that structure-active relationships can be done. So QSAR, as I mentioned here, is a very standard example, standard methodology in chemical R&D, that you can enhance the properties, enhance the binding affinities, enhance the overall drug-like properties of the molecule. So I'm more than happy to dive into this in more detail. But I mean, hopefully that answers your question.
Yeah.
Yeah. Okay. Great. Oh, and others, any questions? I typically tend to go pretty fast, so my apologies on that. But again, I want to give the time to the audience to ask and to stop and ask questions. So, okay, let me go to the next slide. So these are some case studies. We'll give you an example of what Atlantis AI360 can do, right? So this is an example where I took this from Dana-Farber. Atlantis actually goes back that long, but the idea was to take transcriptome level information in HTLV-induced, HTLV-infected cells, and to understand what is the dysregulation caused by the virus, and what are some of the key players involved in dysregulation, right? So, I mean, I took this platform called MPSS, the Massively Parallel Signature Sequencing platform from Sydney Brenner's laboratory when I was at Dana-Farber. And then what the platform gave me was 15 to 20 more transcripts, which were up or down regulated in HTLV1 infected cells. And the question that we asked was, which of these are upregulated in a significant manner? Which of these are downregulated? And can we deduce a pattern? So I had to look at literally millions of these transcripts, and I could have done it, manually. So I utilized my programming skills to automate the whole thing, and in the end, I was able to at least identify a couple of key molecules. So GITR, which is protein member of the TNFR family, was upregulated, overexpressed in HTLV transformed cells. So, on the basis of that, I derived the potential pathway of dysregulation in HTLV. I mean, that is just one example of many, but it gives you a sense of how deep analysis one can do in these kind of scenarios, and how Atlantis really can help you do the analysis much faster. I wish I had Atlantis in a full-blown manner back then, but this was all done, in a program fashion. But Atlantis would have been much more helpful during that time. But again, this is an example of how you can utilize AI Atlantis for whole genome dysregulation kind of scenarios, with just millions of transcripts to look at, and hundreds of different of pathways that you can then ask the question of what is really happening in the pathology of similar diseases, right? Another example is question was, what is the interactome of CDK4 in breast cancer? Not CDK4/6. It is specific for CDK4 only. And based on the basis of NLP analysis, I was able to understand, or at least derive, a picture. I mean, this is not obviously validated, but I can say, for example, with certain level of assurance that with some high confidence that CDK4 may have a relationship with AKT1, or may have a relationship with, let's say, EGFR, or with MDM2. But that analysis obviously has now to be done in the lab, and to prove or disprove that, because this is all in silico. You can only get predictions from AI. But in the end, you have to go back to the lab and either validate or disvalidate or invalidate the hypothesis that there is a relationship between CDK4 and AKT1. But it gives you a clue. And in the end, obviously based upon your own research and based upon your own understanding. So obviously, if you're a researcher in breast cancer, you know exactly some of at least some of the key pathways. And you might say, oh, this is something new. I mean, XIAP or a SMAT3 is a new interaction. I would like to try and do some pull down acids and see if that really is true in triple negative breast cancer. So the idea is to give you enough of a confidence, enough of a workable hypothesis that you can then take to the lab and do some plan some experiments. This is another example of how this can be applied to a real life problem. Another example is more from the biopharma industry. This was a rare disease asset that somebody wanted to license or to acquire. And the question was, how do we know that this is real? So the company wanted to do a due diligence from a clinical KOL, key opinion leader. And so I put Atlantis to use. So it identified a number of clusters. Again, as Sanjay, as you mentioned, these are all team efforts. So I was able to identify clusters in the US, in Europe, in Australia, and as well as in India. And I wrote to a number of people in Australia and in the US, the professor of pathology at Monash, of neurosciences actually in Monash, replied, and we were able to actually do a complete due diligence of the area in 48 hours. So this professor actually was very well versed with the pediatric neurodegenerative rare disease, where this molecule was being used. So again, gives you a sense of how deep dive the Atlantis can do. Identify researchers, identify locations, identify complete geolocation mapping of researchers globally, and give you a clue in terms of who's out there and what potentially we could maybe collaborate with them or at least talk to the KOLs to get the information which is relevant to your field of study. This is another example of how Atlantis was used. Another example is as I mentioned earlier, and this is from AZ. We wanted to understand complete clinical decision tree. So looked at data from Journal of Clinical Oncology and asked a question about what is the typical first line, second line, third line options available for treating various GI diseases. And does AZ have a gap in their portfolio? And so this data was taken by them to define a clinical decision tree and also look at gaps in their portfolio. Another example is, again, this is clinicaltrials.gov. So we're able to quickly identify what clinical trials are ongoing. And from each clinical trial, you can understand, which companies are working on clinical trials, what specific drugs they're using, what endpoints are they using, what molecules are they using, whether it's a biologic or a small molecule. So it gives you a complete, bird's eye view of a large amount of data that you can then utilize for your own research effort. Patent landscape. So, you can identify who has filed patents on CDK4, for example. And again, that gives you a clue in terms of what prior art is there and what has been protected in terms of patents. Again, some examples of CDK4, but again, this is highly customizable. Another example is you can visualization in the and you need to visualize your data. So, for example, on the top left is a clustering data of all the genes involved in epileptic disease. In the middle is the heat map. On the right-hand side is another hierarchical way of clustering the known association between genes and their phenotypic and genotypic effects. Right. So again, the idea is to be able to visualize your data, and Atlantis can help you visualize that data. Because again, in India, you have a lot of text. It is not helpful. When you have visualization, it brings out the relationship associations in a much more interesting manner, and also maybe identify some potential new areas of research. So all of this can be done using which are known as visualization libraries, which Atlantis AI can help you do that. Okay, and so this is all about typical use cases of Atlantis. Again, I'll pause here if there are any questions from the audience.
So this is something new to us, Bal. And
Okay.
so there may not be much questions. We are curious to
Oh,
know
okay.
this material.
Sure. No worries. Okay, so I'll move ahead. So, I've tried to compare, as I mentioned, right? I tried comparing Atlantis with ChatGPT, which is the rage of the day. Everybody was talking about ChatGPT, even Google, right? How is this different, better, whatever, right? I mean, what does it say in terms of the value of Atlantis? And I'll also give you some examples of some demos. So let's dive into it. So this is the user interface for Atlantis. It's almost like a Google like search engine. There are two search boxes there instead of one. You can also put just one. So you can say, hey, CDK4, breast cancer, you search, it'll give you all the data in a PowerPoint presentation, right? This is the overall interface, how it looks like. And in terms of the output, right? The output at the bottom, you see, it gives you a PowerPoint deck. It gives a complete geospatial mapping. of the sites. On the right hand side, gives you the industry view, meaning, is there a BMS working, or is there a Pfizer working on that particular drug or that particular, research area? It gives a complete literature view, right? It'll look at each PubMed article. It'll give you the gist of that particular abstract. It'll give link outs to universities which are working in that area. So UPenn, GWU, Howard University, wherever you have it, if there's data available, it'll give that to you in a click and point, easy to use format. And the bottom will give you much more detailed analysis of a molecular view of the data. So and this is an example of what I mentioned earlier. So it is all very easy to use, as I said, point and click. This is an example of of the question. The Q query here is FXR in steatohepatitis, right? So everything is clickable. So to latest research article, you just have to click it. It'll take you to the particular source. It generates concepts for you, which are known as NLP tags. The On the right hand side, you see it's a fully customizable interface. It links to various top journals. You do access to various institutions which are doing research on that particular topic. If it is there, it'll show it to you. And again, this is completely customizable. This is maybe specific to this particular search, but to your use, it can be completely customized. This is a deeper dive. So it'll look at the abstract, it'll highlight the search terms, it'll identify on the right hand side the deep protein to protein interactions, right? It'll map all of that out, and it'll link these molecules to various websites. So, for example, JAK3, you can click on JAK3 and you see more information about it. It's called Janus kinase 3. It'll show you all the the protein structure, the transcripts, all the information you can at your fingertips. So that's how useful it is in terms of being able to leverage the public data sources to your advantage. So all the search terms are highlighted, very easy to view and to use. But again, as I said, this is the complete geo mapping. So this is for searching the FGFR in cancer. It just shows you clusters in Europe, clusters in the US, East Coast, West Coast, clusters in Japan and South Korea. So, it gives you a sense of, who's out there, where I can collaborate, where I can identify potential patient recruitment sites, maybe even some key opinion leaders who can advise me on, various aspects of that particular research area. And then now this is interesting. I did an analysis with ChatGPT. So if you open up ChatGPT and ask the question, okay, tell me what is the connection between HIV, infection, and aging? It gives you just a text, right? Chronic inflammation, immune system activation, co-infections, antiretroviral therapy side effects, and give you some examples, right, of what could potentially be involved in the pathology of HIV, which causes aging in individuals, right? And then I asked the question to ChatGPT, can you provide a few recent journal articles on the topic, and can you please summarize the results? I was amazed to see the answer was, I do not have access to databases or to subscriptions. Therefore, I cannot provide you specific recent articles, right? How I can guide you. But we don't need guidance. We already know PubMed. We already know so many journal articles. And at the bottom it says here, once you find the relevant articles, you can read the abstracts. Big deal. I mean, that's the basic problem you're trying to solve here, right? So even Chat GPT is not able to solve the problem. So it gives you very high level data, no deep dive into the information, into the subject matter, no molecular level insights, no visualizations. It does not enhance your research workflow. You still have to go manually read those articles yourselves. I mean, that's just tedious, right? It is old school. No, it does not enhance your workflows, and it is just not useful. And yeah, I always keep saying, right, these are all tools are good for uh, industries like maybe engineering, oil and gas, but not in life sciences, where the data is so intense, so complicated, so multifactorial. Same thing with Google. If you do a Google search or Google Scholar search, it does not give you, it gives you access to links. It may kind of store the information for a little bit, but it does not give you analysis, it does not give you insights, it does not give you automation, it does not give you workflows, it does not give you any storefronts. So, that's the beauty of Atlantis. It gives you all of the above. It enhances your workflow, makes your job much more easier in terms of looking at data and really helping you focus on solving, your research problems, your research effort, advancing your research effort. So I'll do a quick demo now, right, of exactly what I did with. So, there was a question about can you, or there was a somebody who registered whose focus was HIV and aging. So I ran a search using Atlantis, and this is what I got. So I'm going to click on this document and open the PPT which was created, which is a PDF form now, but it is a PPT which was created, right? So here is the point and click HTML dashboard. So it summarizes, okay, the top 10 journal articles. It gives you links to the PMID and maybe to the PMCID, which is available. And this is a completely, like I said, point and click. Everything is dynamic. So it gives you the summary. You can click on the links and open each of these papers separately. it now also gives you summaries of each and every paper. So this is the first paper, impact of aging on immunity in the context of COVID-19, HIV, and TB. Right?
Can I
It
ask here
downloads
a
the.
question? Can I ask something? I mean,
Sure, sure.
if you go to PubMed, there is over 5,000 journals that it has HIV and aging. So why there is a top 10? And what is the
No, no,
selection method that it does to select those as a top 10?
no. This is just an example, Alberto. I'm not saying that this is just for the purpose of demo. You can do the 5,000 also. Yeah. So I search to top 10 articles just as a demo.
Okay.
But you're free
Okay.
to do 5,000 searches. Yeah, yeah.
Mhm.
So that you can like do it in a based on year, like, you know.
Yes, yes,
Okay.
yes. Exactly, exactly. Yeah. You can limit to the number of years. You can limit the number of public articles that you want to find. You can limit to the words HIV and aging in abstract, in title, all of the same PubMed stuff. Yeah. Yeah. But this is just a demo. So they'll give you a small, just to tell you exactly how it works, right? Now, if you look at this particular slide, it downloads the name of and this is all hyperlinked. So if I click here, for example, if you want to say, this is interesting for me, right? So I click here, it takes you to the actual source. Right. I'll go back here. So it'll download the title, it'll link it to the particular article, it will download the addresses, the affiliations, the abstract, it will highlight the key terms which have been used for the search, it'll give you the keywords from PubMed, but Atlantis also gives you clinical terms as well as NLP tags. So these are much more useful than the keywords given by PubMed. So it kind of summarizes the article. So age-related changes affecting key components and all of this. So it does much more than what PubMed gives you. Another same example. So it downloads all the articles, all the figures, the images, the graph, the flow charts, schematic diagrams. So you can utilize that to your for your research effort, right? As I mentioned, search terms are highlighted, right? It downloads everything, right? It creates a PowerPoint presentation for you. It identifies the research groups that you can collaborate with. You can derive novel insights based upon this data. You can take this to new directions, right? Essentially what I'm saying is it summarizes the whole search results into a PowerPoint. Now this can be a living document where you can say, okay, this is what is there in literature. I will overlay this with my own results, and that data is now useful for me to advance my research. Right? And of course, if you want to see that paper, you can quickly go here and read the paper itself. Yeah, so some more examples of the same thing, but essentially what I'm saying is that it gives you a complete persistent database of research data. Now, if you want to use it for a symposium, for a presentation, you have it you have it handy. So almost you think it's like almost a 55 slide 51 whatever odd slide deck made in a matter of minutes. Second example is HIV and atherosclerosis. Same, I ran the answer search again using Atlantis, and it gave me this PowerPoint presentation in a matter of a minute or less. So again, the same dashboard, click, point and click, and then all the papers have been downloaded. This can be linked to PubMed, right? So about 20, 30 odd slides. Yeah. So it kind of gives you the power of what Atlantis can do for you. So that's where I kind of wanted to close and again take your feedback.
Hi, I had a question. Can you hear me?
Sure. Yeah, I can hear you.
Okay, so have you limited it to certain databases, or is it going all over the place to get this information?
So this is currently customized to PubMed.
Oh, okay.
Yeah. Because I assume PubMed is the first resource that every researcher goes to. But if you tell me, hey, I need USPTO, I need clinicaltrials.gov, I need other websites, it can be customized.
Okay.
Yeah.
So it doesn't tend to hallucinate?
Well, I don't think so, because this is based upon peer-reviewed data. It will not give you something which is untrue. This is reported, this is peer-reviewed. So that is going. I mean, that's a very good question, and that's how it is different from ChatGPT. Because ChatGPT, I'm not sure whether you can really depend upon what it says, but this is peer-reviewed journals, peer-reviewed articles.
Okay.
So however, now in PubMed, they're listed by archives. journals which haven't been peer reviewed, well, does this platform eliminate those that are in bioarchives?
So I will explain that question. See, Atlantis will not make that editorial decision for you. It'll say this is what is available in the literature. Now, if you are an HIV expert, let's say, for example, you may give credibility to some data and not to some data. You may give credibility to some researchers, not to something. That's your personal call. But I will not hide the information from you because I want all the information to be available to you, and then you can make a decision whether this is the right data or not for you to then pursue. So I will not make the decision unless you tell me I only want high impact factor, I only want Nature, Science, PNAS, JBC, those kinds of articles. I can limit that, but I think you might lose some information which will not be useful to you. I mean, which is not good in terms of your research effort.
So at the beginning, you said also one of the case studies is, for example, to identify interactions with CDK4. Is it the same platform? Like how to
Yeah.
because in here you're looking at the literature, but the other example was just basically kind of a string pathway analysis of, which you can do to find interactions. But how do you come up from to that graph?
So here, so
sorry.
No, so here's an example, right? This is what you're talking about, right? So on the right hand side, if you see here, the proximity analysis. Okay, let me presentation mode. Yeah. So what the algorithm does is that it looks at what particular biomolecules are listed in that or mentioned in that particular abstract in proximity to each other. And then it does a statistical analysis. Okay, is CDK is for example SMAD3 mentioned in reference to CDK4 in 9 out of 10 journals, or it's only 1 out of 10 journals, right? So it does an analysis and says, okay, there is some level of confidence that CDK4 does have a interaction with let's say SMAD3 based upon the known literature, and it gives you maybe a score, right? And that then you take it to your research. But to your question, how does this build the graphic? So we have certain graphical libraries. It takes that data and then it generates that graphic.
It's like the interface seems pretty simple, where you just put 2 words and it came out with a PDF. So based on all the case studies, there is a lot of different things that it can be like
Right.
trying to understand, as an interface, I'm not really savvy on bioinformatics. That's one of my many weaknesses, but I've used a lot of pathway analysis tools, Ingenuity or string pathway analysis, or all these that are available, right? That they kind of do a similar thing where they are building into what is known of information to create, big data analysis similar to that. So is the user interface easier to analyze? Because sometimes the problem with these, what I have, maybe I'm speaking by myself, is that suddenly you have too much information and then you don't know where to look at.
Correct.
So how is all the process, for example, from what you show us that is just basically putting into words to get to all the even generic resources, how do you go through that?
Right. So this is the interface that gives you access to all the abstract, as I mentioned to you, right? I'll show you that PowerPoint, right? So that's how it does. Now we also have something which is called a concierge service. So for example, if you tell me, hey Harsh, I want a deep analysis done into, let's say, CXCR4 in relation to HIV, right? Or just to give an example, right? So we take that particular use case and generate information for you. This platform will not generate that particular as of today, but we can take the information to you from you, develop that deck for you, and also generate the graphics for you. But currently, it does not. I mean, once you do a search, it does not give you that graphic automatically. It will give you the PowerPoint presentation. But now we can go to the next level of doing an NLP analysis and giving you generating the graphic. That's a concierge service that we provide to you. And in the future, I want to make it self-service. So people who do this search will also be able to generate those graphics. I mean, it will require some intervention because in the end, it's a machine. So you need to provide some direction to the machine, but in the end, it will do it for you. Right now, we offer it as a service for you.
Yeah, thank you. That was my,
Sure.
I'm not as savvy on bioinformatics, so I like when some things are easy and it's done.
Sure.
You put your genes of upregulated and regulated, and it can generate you all that nice graphs. So I was just wondering how you get from one to the other. But
Yeah,
then,
you're right. It's all about data. So once you have the data, you plug it in into the appropriate platform, and it does the second level of analysis for you. As I said, the
I mean,
visualization, which are then
and then the search, I mean, is based on PubMed, which is it's good, it's peer review journals, but you are missing like probably there's like information from patents that are not published yet or other. So my question is like you showed and then you limit it to 10, right? But it's like, how do you know what is, like because if you like, let's say HIV and aging, there was 5,000 journals. So if you get a 5,000 journal PowerPoint, that's like the same as going to just PubMed and one by one, right? I mean, it's kind of like,
yeah.
how can you like how as a researcher will say, okay, I mean, you may be limited by years, or is there a way to like increase the number of searching that is like to be more specific? Because
Yeah, yeah,
how do
yeah.
you get into like what is the most relevant piece of
Yes,
information out there? Even if it is per review, it doesn't mean that is true.
sure, sure. Yeah,
That has to be confirmed sometimes. So it's like, how do
yeah.
you using this platform can help as a researcher to identify what is the most important for your
Yeah.
research?
So it is a bit of subjective analysis strategy, right? Look, for example, I worked on HIV myself when I was at the University of Rochester, right? So I know, for example, that there are certain pathways which are important, certain molecules which are important. Maybe I'll refer to this work at Dana-Farber. I think that is the HTLV information that I gave you, right? So yeah, here you go. This is example. So I know, for example, that my research suggests that there's a relationship between GITR and HTLV transformation. So based upon prior information, I will utilize that information to build a stringency into my research. So I will say that, okay, let's do a certain HTLV in title, and I will do GITR in title, not in abstract or not in the text, because I know it has to be a paper where it mentions GITR specifically in reference to HTLV. It has to be in the title. So to your question, I can manage my search, I can make my search more relevant by increasing stringency. So where I'm just starting off a new research, I will say, okay, give me everything. So I want HTLV, I want GITR, I want CD4, I want CD25, the whole 9 yard. So you give me 10,000 papers, right? But as I advance in my research, I know for sure that there are certain areas which are much more relevant to my own research because I can't go after everything. So then I increase the stringency and say, in title, I only want to know the last 12 months. So I'll limit it for maybe 12 months. I also limit it to the top journals if I want to. So it is nothing different than, in a sense, how you use PubMed. There's a bit of an experimentation. Sometimes you get the data in the first run, sometimes you don't. Sometimes you have to dig deeper. But I think overall to your question, it's a subjective analysis based on what your knowledge of the data of the subject matter is, and how then you utilize your overall strategy to then focus on those most relevant papers that you think are the most relevant to your research. So stringency is one way how to control it, and limiting it by number of years. You can limit it to number of journals, the kind of journal that you want. Maybe it's a research journal, maybe it's a research article, maybe it's a research review paper, all the above. And Atlantis will do all of the exact above. There is no different than, in terms of course, it will help you generate the information in insights, but, you can do stringency, build stringency in your searches in that fashion.
So I have a very basic question. How we can access this? Is it commercially available or freely
Yeah.
available like Google?
So currently, as I mentioned, it's a concierge service. So I take the input from you. Suppose you tell me HIV and services, run the program for me, give me a 30, 40, 50 page slide deck with all the insights, right? And I'm done. I'm also building it like a Google-like engine. So this will literally replace Google for life sciences. So so the interface that I showed you, the two search boxes, is exactly how it will look when it will go live in the next few months.
Okay. And do you charge fees for that?
Yeah. So there is per search, there's a charge of $2,500. So it'll generate the PowerPoint presentation, it'll generate the HTML point and click tag, it'll generate the visualizations for you. So that's $2,500 per search. and there's a trial available for, let's say, 1 month for $25,000 for the entire lab. Unlimited number of users, unlimited number of searches for that particular lab. $25,000, which is a trial subscription for 1 month. And of course, then if you like it, you can continue at a different pricing.
Yeah, yeah. And so, say we worked with you and came up with a very novel idea, and then we pursued that idea in the lab, and it has commercial value. So, how much of the intellectual property right goes to Atlantis versus
Oh, no, no, it belongs to you. IP belongs to you. You are the client.
Okay,
Yeah, we
okay.
don't take any IP on that. Yeah,
No,
yeah, we are a service provider.
I just want to ask this.
Yeah, sure, sure. It's an important question. Yeah, yeah. Your IP
Right.
is your IP. I don't take any IP. Yeah.
Okay. In terms of the safety, does it grab our search history and use it as for commercial purposes?
No, we don't. No. This is completely confidential. Yeah. Your
Okay.
data is your data. We don't utilize it for anything. Yeah. This is completely, controlled by you. Yeah.
Yeah.
We don't sell this data. No.
Okay. Okay.
We can
No,
make
this
a we
is,
will make a case study out of it, saying, okay, we did this search for GWU, but not reveal any IP. Yeah.
yeah. I'm just asking you for the broader interest of the audience.
Sure, sure. That's very important.
And this is definitely very interesting, particularly, I mean, if I'm working in certain area, I feel confident. I'm not lost in the information. I have pretty much grasp of what is happening in recent days and everything. That's there always. But if I'm moving into a different area, completely different area, like say tomorrow I want to be doing some research with say diabetes. I never did it before, so
Right.
your search engine would be very helpful for me to get educated and ask relevant questions and test those in the lab. So in that
Yeah,
sense,
that
it
is
is
very
useful.
very good feedback. So, Sanjay, you are an experienced researcher. You know everything about, let's say, HIV and all that, right? So you are an expert in that. You don't need much help there. But there might be a graduate student, there might be a postdoc, who's joining in new from a different field. For them to what do you call, gear up, catch
Yeah.
up with that whole science, I think this is extremely useful, right?
Yeah, but it is useful for students. Yeah, I totally agree
Definitely.
with you. The new students who are coming into the lab and starting a new project, this will be very helpful for them.
I mean, for writing the thesis, they do literature surveys, right? I mean,
Yeah,
we can't do it manually, right? And even reading
yeah,
papers, right? I mean, once they know, okay, I want to read this paper, then you can go and actually do the printout. But you need to have that information first to look at. And that persistent format in a PowerPoint deck, I think, is very useful. At least that's what I personally feel. I mean, I use it in my work all the time.
yeah. So on that note, let me ask the students, the students, if they have questions. All right. Maybe they'll contact you later.
Sure, I'll
I
leave
have
my.
a question.
Sure.
Just a very quick question. How you can you do, because you do this initial search and you generate all these files, and now is it possible that you can do sub searches? How you can prioritize those articles that has more impact in terms of the, citation reports or something that, because, you pull out 5,000 papers, I can assure you that you go through the first 10 pages and you are exhausted already of reading all
Absolutely.
this. And today
Yeah.
there is such a big proliferation of journals that, it's really impossible to narrow your more relevant literature. This is where
Right.
I believe that if you have also a sub engine that you can apply, that can do that search on the
Yeah.
most relevant publications.
Yeah. No, again, very
What
excellent
are your
question. And
thoughts about this?
yeah. No, I mean, I completely agree. So much of data. How do you really look at really the good, valuable, reliable data? So what Atlantis does is that when it looks, when I said, right, so it gives you the title, then the names of authors and the affiliations. So for example, that University of Rochester is well known for, let's say, HIV, or the GW is well known for, the CFAR is a very credible organization which has a long history of doing excellent HIV research, right? So it takes that information and is able to understand, obviously based upon, as it does a search, it also takes into consideration where the publication is coming from. So let's say you have 100 papers coming from CFAR and you have 5 papers coming from, I don't know, I'm just taking an example, coming from, let's say, some lab in India. So the credibility of that is, I mean, it's not in question, but you have a what do you call, a comparison. 100 paper from CFR and Nature, Science, PNS, JB, JBC, and you have one paper coming from a very small institute, which might be relevant, but it gives you that comparison. So I think to your question, what we can do is to add that level of confidence building, that out of these 100 papers, the top 10 which have the credibility, the impact factor, are these top 10, based upon where the researcher is, what is their backgrounds and histories, and all of that can be built in, because information is available on PubMed. Yeah.
Okay. Thank you.
Sure. So yeah, as of today, Atlantis is available for adoption. More than happy to provide. If you tell me, hey, these are some of the keywords which you really want to run, I'm able to generate that PowerPoint for you. And this is not, as you know, right? It is not that you do a search once and it's done. More than happy to work along with you to really give you that insight and that value that you need. And it might take a few iterations, which is the nature of the beast. So more than happy to work with you to do this. So as I said, it's available as a concierge service. I'm also working on the Google-like interface, which will be available in the next, let's say, a few months or so. And then that's a different model altogether. But currently it is available. Also, as I mentioned, right, storefronts. And let me not forget that the very important concept of a storefront. As I mentioned to you, you may have a certain, like you said, Sanjay, right? You may have a small molecule which is very interesting in, let's say, HIV or cancer. And they might be a BMS or a Pfizer which is looking to acquire, because the industry is suffering today. They don't have pipelines. They're acquiring companies which have pipelines, which have molecules, right? So if you have, let's say, a phase 1 or a preclinical molecule which is looking really good in animal models, you should be able to evangelize that discovery to a potential acquirer and say, hey, you can earn license fees up to millions of dollars. You can have a stake in the company as it takes it to clinical trials, and you might get a piece of the pie when it hits the commercial. So all of it could be an antibody, could be anything, could be a protocol, could be a new technique, could be a new algorithm, could be a new process. But you should be able to monetize that using storefronts. So storefronts can be created with Atlantis.
That's exciting. So we are almost at the top of the hour, or a little past that. Thank you so much, Harsh. This is really incredible. And we really learned something new today. And I hope our members will contact you later on individual basis and take it forward. But thank you so much for being available and sharing your thoughts with us. And this is a fantastic journey.
Welcome. You're welcome. And thanks again for the opportunity. I think Vivian will share my coordinates with you. So feel free to reach out to me anytime. You'll have my email ID. So I'll be available and most happy to provide any support that you need.
Yeah.
Thank you.
Thank you. Thanks,
Yeah.
everyone.
Thanks, everyone. Have a great day. Thank you. Bye-bye.
Thank you very much. Bye-bye.
Bye-bye.